So just be safe with them. You know, try not to break them. And also, there's a bunch of like really delicate little parts in here that if you're moving this around a lot or vibrating it, these parts can get dislodged. You see on the bottom of the base, there's actually these rubber feet. So if you're working with a lab partner, you don't want to slide the microscope on the table over to them to look at the specimen once you get it in focus. You either want to pick it up with two hands and bring it to them, or just have them come over and look. That's the easiest thing to do, because then you know, especially when you did all that work with the fine knob at your 100 objective, 100x objective, you know that it's not going to need to get focused again. Just have them come over and look. But these little rubber feet at the bottom are pretty nice. Like, I work in a pretty old building, and if somebody is walking around or they're using a big piece of equipment somewhere in the building or doing construction, which it seems like they're constantly doing, that rubber will help so that the microscope isn't vibrating a bit. And that's frustrating because that just negates all of the work that you did to get that specimen into focus. Now, sometimes, let's say for example, on this slide, the specimen is over here, but your lens is looking right here. Well, you're going to have to move that slide over, you know, to the right or to the left, so that your specimen, which I did in red, gets underneath your lens where the X is. And you won't be able to actually move the slide itself, but this entire stage can be moved from right to left. That's called the mechanical stage. That's where the slide sits. And you move that stage by turning this knob. This one faces downward, vertically. The other two face horizontally. And the stage control lets you move the stage from right to left. So we've got the slide, and we put that on the stage, but we want to hold that in place also. So I've got one clip that's going to be stationary, and we've got another clip that works like an arm that you can move out. So you open that one up, you slide the slide in there, making sure that the stationary clip is holding that, and then you'll release that clip and it'll hold the slide in place. So those are the slide clips. And we need some light with this also. So when you turn on the microscope, the only thing you're doing is turning on the bulb. And then we want to focus that light coming from the bulb onto that slide. And we do that with this guy right here. It's called the condenser. Now, you don't have to do anything to it, but the condenser is what's going to focus that light on the specimen that's on the slide being held on the stage. Any questions with the parts of the microscope or how they're used? Thank you. Okay, so pretty easy, and I know, you know, looking at it compared to actually using it is going to be completely different, but, you know, if you remind yourself of the steps, you take notes, you look at a YouTube video before you actually walk into the lab or your clinical assignment, you'll be able to have a pretty easy go at it.
Now that you know the different parts of the microscope, let's talk about magnification. Magnification of the ocular lens is usually 10x. This means that if you were to view a slide with only the ocular lens, the image will be magnified 10 times. When calculating the total magnification, you must multiply the magnification of the ocular lens by the power of the objective lens used to study the specimen.
All right, I had to see if I had any messages. My family is away. So remember, just multiply that objective lens by the ocular lens magnification and you'll get that total magnification calculation. Now, we do stain our slides, and you'll probably, especially initially, be given a slide that's already prepared, meaning you've got the slide, the specimen is on there, it's been stained, and then there's a thin piece of glass over that specimen, not over the entire slide, but usually just right over that specimen. It's a little bit smaller. So if you have a slide, and they're rectangular shaped, if your specimen is right here, that will be fixed to the slide, and you'll have a thin piece of glass over that. You won't be able to really see it, but it is going to be there. But we've got to stain the slides, because if not, essentially what we're looking at is going to look like cellophane. It's just going to be completely clear looking at. You may be able to see some slight shade, gray shade differences, but you really want some of these things to pop. And staining cells will do this. So whoever's preparing this is going to stain this with two types of stains, an acidic stain and an alkaline stain or a basic stain. And certain parts of the cells are going to be picking up these different stains, and then you'll be able to see the outline of the cell and different structures that are found inside of the cell.
The most basic reason that cells are stained is to enhance visualization of the cell and or cellular components under the microscope. Cells may also be stained to highlight metabolic processes or to differentiate between live cells and dead cells in a sample. Hematocillin is a basic dye that stains acidic structures. The resulting color is a purple-blue hue, and structures that are targeted with this dye are named basophilic. Basophilic structures include DNA, ribosomes, RNA, rough endoplasmic reticulum. Eosin is a Connor stain done after hematocillin and is an acidic dye that targets basic structures. The resulting color is a pink-red hue, and structures that attract eosin are called eosinophilic. The cytoplasm is an example.
And, quite honestly, even though I'm not a microbiologist, I do think that some of these slides can be pretty beautiful, the way that they're picking up these different stains. Now, in a couple of weeks with Professor Bowen, you're going to look at the integumentary system. When you get into anatomy and physiology 2, you're going to look at blood. When you get into the nervous system, which I'll be back for, you're going to look at nerve cells. But as we stain these cells, you can really start to see their individual structures. This one you won't look at. This is bacteria, but that you would when you get into a microbiology class. And, really just to emphasize the importance of the microscope, without having these microscopes, we wouldn't be able to see at the tiny level all of these different tiny structures that are either invading us or just out in our world. So it's a pretty cool video. It's pretty short. There is no voiceover on this. It's closed captioning, but no voiceover. You're going to hear music in the background, but it really just emphasizes all of the really cool things that we can see with a microscope.
Okay, as I watched the video, but I'm so bored. And I'm in class. Yes, before you ask, wondering why is she live in class? Honestly, because I can. Also, I have nothing else to do. You feel me? But if you have anything to ask me, ask. I will try my best to answer while I'm in class. But also try not to be distracted. If that makes sense. I don't know if it does. I hope it does. Okay.
Let's get that out of there. Any questions with how to use a microscope? Different parts, any of that stuff?
No.
Then let's go to the breakout rooms. Make sure everything looks the same, everything was described relatively the same. And when you get back, I'll see if there are still any questions about the microscope. I'll see you guys in just a couple of minutes.
What's that?
Hi.
Yeah, I didn't hear her say anything other than just go to the breakout room.
This is awkward.
I guess we'll just stare at each other till we're out of here.
Right, we gotta get to know each other, ask those weird icebreaker questions.
Oh man.
Well, where y'all from? I don't know. Icebreaker question. I'm Jennifer. I live in Dallas.
I'm DeAndre. I'm currently in Vegas.
I'm Krisha. I'm from Austin.
I'm Olivia. I'm from Utah.
Ooh.
I'm Jovette. I'm in Dallas.
I'm Kat. I'm from Vegas.
Is it as hot in Vegas as it is in Texas?
Mm-mm. No way.
Let's see.
That's what?
It's 99 degrees right now.
Oh, so it's a little cooler right now.
A little cooler, yeah.
Like that. It's 90 degrees here too, which has been pretty warm for here. So,
What part of Utah?
like Salt Lake areas, like northern.
I was born in Salt Lake.
Really?
Yes.
It's fun. It's a fun place. I go to concerts down that way all the time.
Oh, nice. Nice. Nice.
This is you guys' first or second semester?
It's my
First for me.
First.
It's my first, sorry. Do you guys find that these Zoom courses are helpful, or like, or do you feel like it's like tedious, that it's like stuff that we do and then like we have to go back over it?
It's helpful, but then again, also tedious.
I have different classes I think would make more sense to have a Zoom. Whereas like I feel like we're very quiz or just like test oriented in this class. So I feel like I don't need it personally, but I get like I get how it's helpful too. Just cause like I've done like all of high school for me was like through COVID and stuff. So it's just like I already use Canvas and known how to use all this like the online aspect of the class. So it's just
I get it. I mean, a lot of times I feel like, when I'm going over things, I'm like, oh, I can use something, but some days I'm like, okay.
or yeah, like I work nights and so it's like when you have to do something before work or like right after, or I can't. It's too, the timings are just horrible. Like, I just want to sleep.
I also work nights, so I get it.
That is definitely the headache of schooling, is trying to balance working full time as well.
Yeah, I'm a school teacher at the moment, so I'm actually on break, but we go back on the 4th. So we start, and then our kids come back on the 12th. So I'm gonna be doing a lot. So I went from teaching science last year, last 2 years. Now this year I'm gonna teach math and science. So I have to learn a new curriculum and do my schoolwork too. So I'm gonna be pretty jam-packed.
No, that sounds fun. I mean, a lot. It sounds like a lot, but it does sound fun.
It's gonna be a lot, but I mean, hey, what's life without a challenge, right?
Exactly.
Yes. I was trying to see how I can condense some of my classes from.
I'm trying to condense work.
Yes. So, like, I have a master's degree. So I have a undergrad in healthcare admin and a master's degree in healthcare admin also. But Galen only took like 18 credits out of all of that. So.
Yeah, I was originally doing ultrasound, and I was doing that for a year and then transferred over. So they took like 20 though, 20 of mine. So I was surprised on that.
Yeah, but it's still not like enough because you need like 144 credits here. So I'm like, how can I like move some of these classes over? So I think next semester I'm taking like 16 credits.
Yeah, that's a good chunk.
Oh yeah. I mean, well, because I will only have like one Zoom course. Everything else would be like at your pace. So like it'll be pretty easy. Because I went to University of Phoenix, so paper writing is nothing to me. I can put a paper together real quick. No, that's
the only thing I hate doing is paper writing.
Yeah. Well, I don't normally type them. I just kind of like look at the information on the screen after I read over it, and then I kind of like just top text it, and then I take all of that, and then I put it on the screen, and then I edit the paper. So it kind of makes it a little bit easier, and plus you're putting it in your own words, and then you're able to insert your citations, where you.
Never thought of it like that.
Yeah. Because I mean, think about it. I'm not a fast typer, so
Yeah, voice texting or like like typing saved my life. I love that feature.
absolutely. So,
Because I think it a lot faster than I can, type it out. So I'm just like, I don't know. I lose that train of thought so easy, and then I'm like staring at the paper. Like I wrote half this sentence, and now this other half doesn't make sense because I've skipped ahead of myself.
absolutely. So, like, if you're just literally, I don't know. It's a way I've learned how to do it because it just makes it easier. Because every time I would type a paper and then I would send it through the plagiarism, I'm like, and then you have to redo it and da da da. So I learned how to like kind of finagle the words as you're like reading. You kind of like change the words around, and then you're still citing. It's still the information. You're just citing your rewording. So I hope that helps some of you guys or in writing in the future.
Yeah. I feel like we got it though.
Of course.
Good enough.
Hi.
We got it.
If I look distracted, I'm writing discussion posts.
No worries. I'm my laptop doing math stuff. So
Some of these discussion posts are annoying. Not
I
even gonna lie.
hate discussion posts.
Can you guys see my screen?
Yeah.
I think we're supposed to do the prop.
Oh, okay.
So I guess we just have to match them up.
See, but you didn't even say that. Like,
So the ocular lens is A, right? Oh, we're about to leave the break room. Let's try it at least.
these gonna be your. I think it's going to be your
Nose piece?
focus. No,
Yeah.
that's G.
Oh,
G
that's-
nose piece.
G is nose piece.
Yeah.
B,
F is the base.
F is base.
B is the lens. The other lens.
Objective lens.
Yeah.
E
is the
Slide.
Is C the slide?
Yeah. Oh.
This meeting is being recorded.
Closed. How did that go? Everyone should be getting back now.
Good.
Okay, good. Then we've got let me get over there. And we've got the cell membrane. That's the next thing that we're going to get to. So the cell membrane is a really important part of the cell. I kind of like to think about it as like the skin of the cell. Our skin is a barrier to the external environment, keeping things outside that we don't want, and then also keeping things inside that we want to keep. But it's selectively permeable, right? It's not that my skin doesn't give anything off or it can't absorb anything across my skin, but it is selectively permeable, and we don't want everything just like going in and out of our skin all the time. Like I can go into the pool and I'm not going to soak up the water like a sponge. But if I sit in the pool for a long period of time, of course, there's going to be some water that starts to accumulate through my skin, right? We can see that because the skin starts to get white, very, very pale, or it starts to get wrinkly or things like that. Plus, our skin, this can help us to interact with the external environment. We have sensation coming from some receptors there, and we'll see that with the cell membrane as well, is that the cell membrane is going to help us to have some type of, have those cells have some type of reaction or sensitivity to its environment. So that's why I kind of liken the cell membrane to our skin. Now, with the cell membrane, there's a few different functions that we'll have to know. And because we're still at the molecular structure, especially, I like to show a few videos so somebody can describe it once, I can describe it a different way, and then you can read it in a different way. And hopefully that hits all of the learning types that we have. So watch the Amoeba sisters. They're going to introduce the cell membrane.
In my second year of teaching, I was looking to improve an osmosis lab I had done the year previously. Osmosis, if you remember from our osmosis video, involves water traveling through a semi-permeable membrane, potentially a cell membrane. And I wanted a cool way that students could model this in different scenarios. And one of my colleagues told me about this egg lab. I won't get into the whole lab though. It was actually one of the first steps of the procedure that got to me. To get ready for the lab, my colleague told me, you can soak eggs in vinegar for 24 to 48 hours and the shell comes off. Oh, so I need to make hard-boiled eggs then. No, raw eggs. But if the shell comes off, well, that's the whole point. What's underneath the shell is going to mimic a cell membrane. It's kind of modeling how a cell membrane would function. You know, if the whole egg was actually like a cell. So then you run different scenarios with it for your osmosis lab because it will be a semi-permeable membrane, like a cell membrane. I couldn't visualize this. If the shell comes off, but it's a raw egg, how does it stay together? So I have this area in my house that's designated for things for me to try out. Don't worry, I always clean up afterwards. I tried this experiment out in advance, just to be sure. The hard shell is removed, but the membrane that was always there remains. We often visualize the membrane of a cell this way, like this membrane around the chicken egg. The cell membrane is semi-permeable, meaning it lets some materials through, but not others. We have an entire video all about cell transport and how materials can pass through the membrane. A cell could never be as large as a single chicken egg, though. Why? Well, it turns out surface area is a very important thing. Remember that surface area determines the surface measurements of that cell membrane, and the cell membrane controls what goes in and out of cells. That includes food coming in, as well as molecules that are essential for metabolic processes, and then also waste going out. If volume, which is all this space inside, increases, then you will have more need for surface area, as you will have more need for food to enter, more of a need for waste to be removed, and more metabolic reactions occurring in this larger volume in the first place. If we do a little bit of math here between these two models, I'm going to use popular cube models instead of egg-shaped models because it's a little faster for me to do surface area and volume calculations. See how here there is a big difference in surface area to volume in this smaller model? 6-to-1 ratio. That means the surface area in this small model is 6 times more than the volume. Look at this beautiful ratio with so much surface area. But if we look at this bigger cube and we do some math for this model, that surface area to volume ratio decreases. Sure, the surface area is still larger than the volume in the large model, but it's only 2 times as large now, not 6 times as large. Cells are way smaller than this small model here to allow for an exceptionally large surface area to volume ratio. And a major reason why we're not going to find a cell as big as this chicken egg here. Surface area is important. And while we can model a lot of the processes of cell transport from this egg membrane and how important the membrane is, I don't want to neglect the membrane structure itself, because the cell membrane structure truly is magnificent. And since every single living thing is made up of one or more cells, which is part of the cell theory, it's a big deal because every single cell has a membrane. It doesn't matter whether you're talking about bacteria or protists or plants or animals or fungi. Even archaea aren't too cool to have a membrane. They all have a cell membrane. The structure can vary some, but we're going to be talking about some major structures of the membrane that you can actually find in most cells. We should mention that the fluid mosaic model is often what we use to describe the cell membrane. A mosaic, in case you've ever created one, we did in some of our art classes over time, arranges many small pieces together to make some large piece. You'll see that that makes sense when describing the membrane in a minute. The word fluid implies movement, and this is true for the cell membrane as the components are floating around, they're not static. So let's take a look at some of these components in the cell membrane. We're looking first at a phospholipid bilayer. A phospholipid is a lipid, but an interesting one. So when you talk about a lipid in general, many lipids are nonpolar. Think of oil, for example. It's nonpolar. It won't dissolve in water. Water is polar. But a phospholipid is interesting because one part of it is polar, the head, and the other part of it is nonpolar, the tail. It's amphiphilic. Let's explain what we mean. We often refer to the polar head of the phospholipid as hydrophilic, which means that part loves water. Well, you know, if a lipid could love. The nonpolar tails are hydrophobic. They do not like water. These phospholipids arrange themselves into a phospholipid bilayer with the nonpolar areas here in between, away from any water. It also allows this area in between to be separated from the outside and inside. Water can be found on the inside and outside areas. Also, these phospholipids, they don't just stay put. They move around. It's the fluid mosaic model after all. This gives the cell membrane flexibility. Phospholipids can even flip-flop around, but that's far less common. Remember that this entire phospholipid bilayer borders the whole cell. It would be a sphere, even though we're just looking at one area of it. We have an entire video that talks about which molecules can go in and out of this membrane and which ones can't that you can view. But for now, we're going to look at some of the other structures more in depth. Cholesterol. You know, cholesterol often gets a bad reputation. And while cholesterol that builds up in the arteries can be a problem, cholesterol in your cell membrane is critical. If temperatures drop, the cholesterol can actually function kind of like spacers between these phospholipids, keeping them from becoming too packed. Or vice versa, the cholesterol can actually function to connect phospholipids, to keep them from becoming too fluid in warm temperatures. Proteins. In protein synthesis, we talk about why it's so important for cells to make proteins. Many proteins are found in or on the cell membrane, and they play major roles. Peripheral proteins, like the name suggests, tend to be on the peripheral area of the membrane. So while they're on exterior areas of the membrane, they generally are not going to go through the membrane. That's for integral proteins. Integral proteins go through the membrane. Oh, and peripheral proteins can sit on them sometimes. Because of location, these proteins tend to have different functions. Integral proteins, with their potential to go through the membrane, are frequently involved in all kinds of transporting methods for all kinds of materials. Some relevance? Consider the breakfast you ate this morning. Your body digested what you ate for breakfast to obtain glucose. Once in the bloodstream, those glucose molecules can't just squeeze through the phospholipid bilayer to enter all of your cells. The glucose molecules are too big and polar, but your cells need glucose to survive, to make ATP, and they rely on integral proteins to get it. Peripheral proteins tend to be more loosely attached, since they're generally not stuck in the membrane. They have an assortment of functions, such as acting as enzymes to speed up reactions, or attaching to the cytoskeleton structures to help with cell shape. Both protein types can have carbohydrates bound to them, which can make them considered a glycoprotein. If the carbohydrates attach to a phospholipid, you could have what's called a glycolipid. Glycoproteins and glycolipids can identify the cell as belonging to the organism, self and non-self recognition, which is very important when you're fighting pathogens. They can also be involved in many kinds of cell signaling. In fact, here's some relevance. A glycoprotein known as CD4 is found on the surface of some of your immune cells. The CD4 glycoprotein is essential for some of these immune system cells to interact with each other and activate. However, it's also exploited by the HIV virus. The HIV virus uses that CD4 glycoprotein as a way to bind to helper T cells, which it then can infect. Understanding the components of the cell membrane and how those components are involved in recognition and cell signaling is critical to understanding how to fight back against many viral and bacterial diseases. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so hopefully that gave you a good idea of what we're going to look at with the cell membrane. So that function of the cell membrane, again, it really is to act like a barrier, but it's a selectively permeable barrier. You know, again, some things can get in and some things can get out. We do want to have an exchange of substances, but we want regulation. Now, I apologize. Sometimes when I talk about this, I keep going back and forth between saying skin and things like that, but I know perfectly well we're on the membranes yet. We're not on the integumentary system yet. Your first system actually comes up in a couple of weeks, but I've got extracellular fluid on the outside of the cell, intracellular fluid on the inside of the cell. And also these cell membranes, because of the glycolipids and the glycoproteins that are hanging off of the edge facing the external environment, we can have some sensitivity to the external environment of that cell. But also we want some structural support. We don't want this thing to just flop down. So looking at this picture to me is a little frustrating, because when I scroll over the membrane lipids, they disappear after a short period of time. But the major type of our lipid protein that we have, let me see if I can get it, is going to be the, all right, I did, phospholipids. That's the main type of lipid that we have. And the phospholipids are what we call an amphiphilic molecule, meaning that it's going to be both water loving and water fearing. So those circles that I just drew, well, those are the phosphate heads, and they're hydrophilic. They love water. So they're going to face the external cellular fluid environment and the internal cellular fluid environment. But we know fats are not going to be water soluble. They're hydrophobic, right? So the fatty acid tails of the phospholipids are going to be facing each other. And this is what gives us that phospholipid bilayer. Now, that's the majority of the components of the cell membrane, the majority of the molecular structure of the cell membrane. But we also have other lipids. We have glycolipids. Make the word make sense. Glyco is just sugar and lipid is fat. So this is a sugar hooked up to a fat, not that big of a deal. And then we have cholesterol also embedded, and cholesterol is a type of a lipid. Now, if the phospholipid bilayer is there to give the cell a little bit of fluidity, well, the cholesterol is there to give that cell membrane a little bit of rigidity. I kind of think about the phospholipid bilayer as helping to make sure that if these two cells, if a couple of cells kind of bump into each other, they're not going to completely shatter like two glass ornaments or something like that. Plus, you know, we can kind of squeeze this stuff around a little bit, but I don't want it to be too flimsy, and that's going to be the job of the cholesterol as well as the cytoskeleton and a couple of other things. And then we've got some membrane proteins. These guys are either just going to be on the outside or on the inside, and we would refer to those as peripheral proteins, or they're going to completely go through that cell membrane. Part of it will be on the outside and part of it will be on the inside. This is an integral protein. This is a transmembrane protein. And a lot of times, but not always, these transmembrane proteins are going to work as channels, like doors to get in and out of that cell. You know, some things can just diffuse right across that phospholipid bilayer. Some things have to go through those doors. They have to go through those channel proteins. So here we are looking at the fats again that are embedded in the cell membrane. Cholesterol. This is an interesting structure because these are ring-like structures. Steroids are a type of fat that we have, cholesterol being one of them. We also have plant sterols, not a big deal right now, but these are what is going to be found in that membrane to help to give us some, not only rigidity. I don't want to make it sound like this makes it really, really hard, because we don't want that, but it makes it so it's not completely flimsy, and then also is going to help so that the cell can, you know, face some of these changes that are going on, whether it's a temperature change or other internal changes that are occurring. And our phospholipids, well, that gives us this amphiphilic structure. And that term amphiphilic is not here, so. Amphiphilic means it is both hydrophilic and hydrophobic. And then we have those glycolipids, just a sugar that's attached to a fat. And what these guys can work on doing is they can function as a recognition for that cell. So our immune system is going to go around with the immune cells, white blood cells. You'll talk about that in anatomy and physiology too. And they're going to look at cells and they're going to try to figure out if this is a cell that should be in our body or if this is a cell that shouldn't be in our body, a foreign cell. And our glycolipids and our glycoproteins are going to work as these cell identification markers to let the immune system know, hey, this is a self cell, you can leave me alone. But all cells, bacteria cells, Brett's cells, Stacy's cells, everybody's cells have their own unique glycolipids and glycoproteins on them. So if I get a cell from Brett and it's not supposed to be in my body, like a red blood cell or something like that, my immune system is going to recognize that and attack it. So it's really for this identification marker. Should it be there or not? These membrane proteins come in a couple of different flavors. We've got the integral proteins, again, going completely through that membrane, a lot of times working as a channel so that structures that are hydrophobic can get through. And then the hydrophilic structures can get through. So we can have these pass in and out of the cell. Or there'll be peripheral proteins, peripheral on the outside, right? So either on the outer or on the inner side. And a lot of times these will work as cell identification markers as well, or they might work as an enzyme. And we saw last week what the functions of enzymes are. Sometimes these enzymes are on the outside of the cell membrane or the inside of the membrane. And remember, all enzymes are proteins, and this would cause a chemical reaction to occur.
In this video, we will be discussing the structure of the cell membrane. When scientists looked at the selectively permeable cell membrane, they described its structure as a fluid mosaic. You might know that a mosaic is a picture made up of little tiles. Like a mosaic, the cell membrane is made up of different parts as well. The cell membrane has two layers of phospholipids, referred to as a lipid bilayer. The lipid bilayer isn't rigid. The phospholipids in it have the ability to move in a flexible, wave-like motion. Let's take a closer look at a few phospholipids. The round head portions are hydrophilic, which means they are attracted to water. Both the extracellular fluid, meaning fluid outside the cell, and the cytoplasm inside the cell are mostly made up of water. So the hydrophilic phospholipid heads of the outer layer will be oriented toward the extracellular fluid, and the heads of the inner layer will be oriented toward the cytoplasm. The phospholipid tails are hydrophobic, which means watery areas repel them. So they orient toward each other in a direction as far away from the watery content as possible. There are also scattered proteins embedded in the phospholipid layers, some with carbohydrates attached. So, in the fluid mosaic model, the cell membrane is made up of different parts. And these parts make up a flexible boundary around the cell. But how do the majority of substances get in or out of the cell? Some molecules seep through the little spaces in between the phospholipids, which make up the majority of the semi-permeable cell membrane. However, other molecules are too big to fit through the cell membrane this way. So how do these larger molecules pass through the cell membrane? The molecules move through proteins embedded in the cell membrane, either from the extracellular area into the cell, or from the intracellular area out of the cell. These substances will move through tunnels made up of these proteins. We'll explore how things move through the cell membrane in greater detail separately.
Okay. Any questions on that cell membrane? Again, just a really important structure that we have. If there are no questions with that, take a break. Come back at 20 after, and we'll pick up with the organelles. I'll see you guys in a little bit.
Thank you.
You're welcome.
Oh, finally I get a break. But it's only like 11 minutes. So, yeah, there's that. I missed my morning class. I woke up like an hour early. And I was like, bro, I still have an hour. Let me just go back to sleep. And then I woke up an hour after class started. And I tried to get in, but they wouldn't let me in. So I messed that up. I don't know what I'm gonna do anyways. But on the bright side, I made it to this class. But the other class is more important. And if I miss one more class, they'll kick me out of the class. So I can't risk that. Ever. Which means from Monday, I have classes mine is on Wednesday. So from Monday, if my alarm goes off at 12 and I wake up at 12, I should wake up at 12 and stay up until class starts. Maybe even, study. Anyways, in the meantime, while we're having this break, let me finish writing down these notes. So by the time she comes back, she's talking, I'll be doing my discussion. I hate doing discussions. I hate. And they say, oh, hate's a strong word. No, it's Junini. I hate. I'm so tired. My watch is almost full.
In this video, we will be discussing the structure of the cell membrane. When scientists look...
I already watched this video. Okay, let's label this. Let me show you guys. This can go for now. Okay. Oh, is that cholesterol? Okay. Extracellular fluid. I don't remember seeing this. Yay! Nice. Okay, now that I'm done with that, let's go back. So the 500 error, what's that? Bro, what's happening?
Okay, so it's 20 after and we're gonna be picking back up. And now we're gonna turn our attention to the cell structure itself. And the different organelles that we have inside of the cells. So these organelles are going to literally mean little organs. That's what this means. And each one of these organelles, like the organs of our body, have their own function, right? Like our lungs, they're there for gas exchange. Our stomach is there for digestion. Our ears are there for hearing. More detail will be coming up later on. So it's important to understand what each one of these organelles are going to be doing, because as you see, these different types of organs that we have when we get to the organ system, you're going to note that, like the examples that I gave you before, my liver cells, my hepatocytes, are going to contain more of this particular type of organelle in their cells, in the hepatocytes. Hepa is liver. So that's why my liver is really a big player in detoxifying. Or my skeletal muscle cells have an awful lot of mitochondria. And that's why I can work my muscles in, exercise and things like that, because mitochondria, its function is to create ATP. And actually, when you condition yourself, when you're exercising and training, and it gets easier and easier, well, really what's happening is your skeletal muscle cells have created more mitochondria, and now you have more mitochondria in your skeletal muscle cells. So it does make it actually easier to exercise. I'm gonna share my screen. I did see a chat in here. Sorry, Jennifer, I don't see the chat when I'm sharing my screen. I'll answer that now. Yes, assume, Professor Bowen, assume I am not here. Okay, so Professor Bowen is taking over the course completely, okay? If I can get in, I will, and I'm gonna let her know. I'll help her out when I can, but assume she is doing everything, because remember, I'm out of the country, so technically I'm not supposed to be able to get in. I may have a backdoor way in, but.
I just wanted to make sure that we didn't have to wait for our grades to come back for 3 weeks.
Oh, no, she will completely take over the course. She's going to do the attendance. She's going to do the Zoom classes. She'll answer questions. The only thing she won't be doing is posting announcements. Okay?
Thank you.
Yeah, no problem. So watch the Amoeba Sisters introduce that. Any other questions with that stuff, by the way? Or anything that I missed in the chat? Okay, I'll let the Amoeba Sisters introduce the organelles.
If you had to think back about the most exciting day you ever had in a science classroom, which day would that be? Now, looking back through the years, we have a few. The time we participated in an earthworm dissection, the time we took apart an owl pellet, the osmosis eggs, all of the fruit flies and genetic experiments. Oh, I could go on. But I will never forget one day in my 9th grade science class. My teacher brought in pond water, and I put one drop of pond water on a microscope slide and saw the most amazing thing ever. I saw an amoeba, a single-celled amoeba on that microscope slide, and I was forever stuck on science from that point on. Because I could not believe that this little cell was there, alive on this slide, still eating, because that's what amoebas do a lot. But to imagine that every person is actually made of billions of cells. Of course, not amoeba cells, but animal cells. Billions of animal cells. That's fascinating. In fact, it really makes you reflect on some of the incredible statements of the modern cell theory. The modern cell theory includes the following. First, that the cell is the smallest living unit in all organisms. And second, that all living things are made of cells, one or more cells. The amoeba I observed was a single-celled organism, so unicellular. Humans are made of many cells, so multicellular. And third, all cells come from other pre-existing cells. You know, cells have their own little world inside them. They carry genetic information, they can divide, many have functions and processes that their organelles, structures inside them, can take care of. On our planet, we can divide cells into two major groups. As a cell, you're either a prokaryote or a eukaryote. Bacteria and archaea are prokaryotes, but everything else, plants, animals, fungi, protists, are eukaryotes. Both prokaryotes and eukaryotes have genetic material. Both have cytoplasm. Both have ribosomes, which are small organelles that make protein. Both have cell membranes, which control what goes in and out of the cell. But what makes them different is a big deal. Prokaryote, the pro rhymes with no, and they have no nucleus, which holds the genetic material and controls the cell's activities. Prokaryotes have no membrane-bound organelles. Membrane-bound organelles are fancy organelles like the nucleus and mitochondria and Golgi apparatus. Eukaryotes, the U rhymes with do, they do have membrane-bound organelles. So now you may be wondering, well, what do the organelles do? What are their functions? Well, you know our style. We love our science with a side of comics. So we want to take you on a tour of the ride of your life into the inside of a cell. Now, to start our trip, we're going to have to get through this cell membrane, also called a plasma membrane. It's selectively permeable, which means that it'll only let certain select materials in and out. But by doing so, it keeps things in the cell stable, also known as keeping homeostasis. And we have an entire video on just the membrane itself, which is found in all cells. But for now, we're just going to have to squeeze through this protein in the membrane. Now inside the cell, we find ourselves in this jelly-like material called cytoplasm. It surrounds all these internal cell structures, and you'll find it inside both prokaryotes and eukaryotes. Now, organelles that are floating around in the cytoplasm can have more support than you might think. Cells contain a cytoskeleton, which is a collection of fibers that can provide support for the cell and its organelles. And the cytoskeleton can even play a major role in cell movement. The cytoskeleton actually deserves its own video because it's very complex and its organization can vary depending on what kind of cell you're looking at. Moving through the cytoplasm, let's start with ribosomes. They are not membrane-bound organelles, and they're going to be in both prokaryotes and eukaryotes. They make protein, which is really important because that's what so much of genetic material, DNA, codes for, protein. Ribosomes can be free in the cytoplasm, but they can be attached to another organelle too, which we'll talk about a little bit later. We're now going to focus on organelles that will be membrane-bound. So we're going to be focusing on organelles that you would find in a eukaryote cell. This takes our travel to the big boss, the nucleus. Now in eukaryotes, it holds the genetic material. Genetic material as in DNA, for example. Both prokaryotes and eukaryotes have DNA, but if you're a eukaryote, you have a nucleus to put it in. And the nucleus controls the cell's activities, and inside it, it has a nucleolus, which is where ribosomes can be produced. Attached to the membrane of the nucleus, or nuclear membrane, you will find the endoplasmic reticulum, ER for short. It does a lot of processing of molecules for the cell, like protein folding, and it's highly involved in actually transporting those molecules around, like a highway. And there's a rough ER, which has ribosomes attached to it, making it, as you can imagine, rough, and then smooth ER, which doesn't have the ribosomes. Rough ER specifically tends to be involved with protein producing and transporting, because remember that ribosomes make protein. Now, molecules that leave the ER can be sent away in vesicles that actually pinch off the ER themselves. Now, smooth ER has many additional roles, including detoxification, which is one reason why your liver cells tend to have a lot of smooth ER. And another additional role of smooth ER is that it can make some types of lipids. Next, the Golgi apparatus. It's the ultimate packaging center. It can receive items from the transport vesicles that pinched off the ER. It has enzymes that can modify molecules it may receive and sorts the material it receives as well. It can determine where to send those molecules, including some that may eventually be sent to the membrane so they can be secreted, which means items that can be sent out of the cell. So with all that's going going on in here, you might start to wonder, what's powering this thing? The mighty mitochondria, or mitochondrion, if you're just talking about one. They're like power plants. The mitochondria make ATP energy in a process called cellular respiration. Now, it's not the type of power plant that you might think of. It runs on glucose, which is a sugar, and it needs the presence of oxygen to efficiently make ATP energy. Now, at this point, we need to mention that eukaryotes are not a one-size-fits-all. Animal cells can have differences from plant cells. We have a fork in the road here. For example, plant cells not only have mitochondria, but they can also have these awesome organelles called chloroplasts. Chloroplasts actually make glucose by using light energy in a process known as photosynthesis. They tend to even have a green look to them because they have a pigment that captures the light energy and it reflects green light. Now, both plant and animal cells can have vacuoles. Now, vacuoles can have a lot of different functions, but many types act as storage of materials. Plant cells can have one large vacuole called a central vacuole, while animal cells can have smaller several vacuoles. Now, remember how we already said that all cells have membranes? They do. But plant cells additionally can have a cell wall, which is a layer that offers additional protection and shape maintenance that animal cells do not. Okay, now how do we get out of this animal cell that we've been in? Well, we could get out like a protein would. So if we were a protein, we would only be made because of instructions from DNA. And remember that in eukaryotes, DNA is found in the nucleus. We would be made by a ribosome because ribosomes make protein. The ribosomes could be attached to the rough ER, and the rough ER highway would provide a vesicle to send us to the Golgi apparatus where sorting can take place. And if we're tagged for being secreted, we could be sent off through a vesicle from the Golgi to the membrane. Out we go. Now just keep in mind that in our quick tour, there are still so many more awesome organelles found in lots of different types of eukaryote cells to continue exploring, so to the Google for more. Well, that's it for the Viva Sisters, and we remind you to stay curious.
Okay, just a nice little overview of the cell organelles. And this next picture that I'm going to show you is highlighting each one of those organelles and their function. So I always like to start off with the nucleus because, you know, that's where we have the control center of the cell, really. And the nucleus is the biggest organelle that we have inside of the cell, and it contains our DNA. Now, DNA is not just there for sexual reproduction, and it's not just there to make sure that we can divide these cells and get new cells when cells wear out or need to be repaired, or we start as this little person and get to this big person. That DNA is also going to drive protein production, and different cells create different proteins. Now, we all of our cells of the body have the same DNA, except for our sex cells. More to come with that in anatomy and physiology too, meaning the oocytes and the sperm cells. But what we mean by this protein production and how this is going to play out is that the cells of my GI tract make enzymes, which are proteins, and that is driven by the DNA that's found in those types of cells. But for example, my red blood cells make hemoglobin, which is a protein, and it's a pigment too, but it is a protein that's going to help to carry oxygen around. So even though they all have the same DNA, some of those proteins.
I'm sorry, but I don't understand Spanish. My bad. Can you translate that to English, please?
When we're not going through cell replication, which is really most of the cell's life. And chromatin is basically like if you took some string and threw it on the ground, that's what chromatin looks like. But when we're going to go through the process of cell replication, mitosis, that's when the chromatin condenses and we can see the chromosomes. So chromatin is unwound. chromosomes. Chromosomes is the condensed chromatin that we see right before the contents of the nucleus divide. Surrounding the center of the nucleus is an envelope. These organelles are going to have membranes around them. So we call this the nuclear or the nucleus envelope. Either way is perfectly fine. But we still have to have a way to get things from the inside to the outside. And that's done through the nucleus pores, through the nuclear pores. Now, from the nucleus, what we're going to see is there's going to be another membrane-bound organelle coming off of that. And it's just a continuation of that nuclear envelope. This is the rough endoplasmic reticulum. And we call this the rough endoplasmic reticulum because it has beads on it. And so it makes it look like it's rough, makes it look like it's bumpy. Those little beads are the ribosomes. Now, ribosomes are the organelle that is the site for protein synthesis. This is where proteins are actually created. You know, the stuff that is driving, that DNA is driving, what protein is this particular cell going to be making. And the ribosomes themselves, they are consisting of small and large subunits. And there is a difference in a couple of different types of ribosomes. Some of them are free ribosomes, which means they're just floating around in the cytosol. Others are actually bound to the endoplasmic reticulum, making that endoplasmic reticulum rough endoplasmic reticulum. So because of now knowing what the function of the ribosomes are, we know what the function of the rough endoplasmic reticulum is. This is the location of protein synthesis, where ribosomes are embedded there. So the ribosomes are doing the job, but this is kind of like the place where this is happening. The fixed ribosomes are found here. Now, when those ribosomes on the rough endoplasmic reticulum create their proteins, those proteins go off in a little vesicle, and they make their way over to what's called the Golgi apparatus. And the Golgi apparatus is going to be a place where we modify and then package these proteins up for release. We're going to bring these proteins to the cell membrane so they can help to repair damage that's occurring to the cell membrane wall. Or maybe we're going to package these enzymes up into another organelle called a lysosome. Remember, all enzymes are proteins, but not all proteins are enzymes. And then we also have the smooth endoplasmic reticulum, another membrane-bound organelle, and it's continuous with the rough endoplasmic reticulum. So it's the nuclear envelope, that membrane is continuous with the membrane of the rough endoplasmic reticulum, which is continuous with the membrane of the smooth endoplasmic reticulum. Now, we get the name smooth endoplasmic reticulum because this doesn't have those beads. It doesn't have that rough appearance. So we're not making proteins here, but instead, we're going to make carbohydrates and lipids here. You know, like if I eat too many calories, I make fat. This is where this is occurring. What else we got here? So we got the lysosomes, and these are these little balls, these little vesicular balls that are floating around in the cytosol. And inside of those organelles, we're going to have enzymes. Enzymes digest. So these lysosomes will take up maybe some organelles that are no longer working. They're worn out.
I'm actually in class.
Ingested
I'm in class. I'm in a Zoom class right now.
Fuse those two membranes, and now the enzymes in that lysosome will digest that bacteria. Perioxysomes are similar to lysosomes, but instead of breaking down unwanted substances, the perioxysomes are going to detoxify substances. So inside of the perioxysomes are are enzymes that are specifically going to detoxify. So again, a perfect example of where we're going to have a lot of perioxysomes would be the hepatocytes, the liver cells, where we have detoxification occurring. We have the mitochondria. Mitochondrion is singular, another membrane-bound organelle, and this is where ATP is being produced. So you know all of those calories that you eat from carbohydrates and fats and proteins, well, we don't actually give those to a cell to have a cell do its job, to do work. Instead, we take all of those, we rearrange it and convert it into that magical molecule called ATP, and then ATP can be given to a cell, and then that cell will do its work, do its job, whatever it may be, contraction, releasing some substances, you know, whatever it may be. So the more mitochondria that you have in the cell, the more ATP that you're going to be able to produce. We have centrioles, and these guys are made up of microtubules, basically proteins again. Proteins do a lot of things. And what's cool about these guys is these guys are going to help with that cell division. So we'll see in a little bit when we have cell division occur that the centrioles will go to each opposite side of the cell, and it'll grow these arms that'll pull our chromosomes apart, sister chromatids to be more specific. That's the major function of the centrioles. We have the cytoskeleton, and this basically does what our skeleton does. It gives us this structural support. It's made up of proteins. And also I like to think about the cytoskeleton as like the railroad tracks. Like if I need to move something inside of the cell, I've got this pathway to move this stuff. It helps to transport substances within the cell. And then I'm not a big fan that they call this, or they have this with the organelles, because this isn't an organelle. This is really just like a structure that's kind of coming off of the organelle. It's called the microvilli. And what the microvilli are these finger-like extensions coming from the cell membrane, or that have cell membrane over it. So in the center of the microvilli, we still have cytosol and cytoskeleton as well. But the important function of the microvilli is that it will increase the surface area.
No, they can't hear me. The only one who's able to speak right now is the professor, and everyone else is muted unless, you know, you have to speak. So no, they can't hear me. What do you mean you're going into? Oh, you're going to year 7. What year are you? I'm in college for nursing school. Yeah. Are you the one who lives in the UK but grew up in Dallas?
Less surface area than if I had an undulating line, a wavy line between the starting point and an ending point, which we see in B. Because say, for example, I take that blue line in example B and I spread it out. Well, now maybe that distance is from here to here. So you can see that I've got all of this space for absorption to occur, right? A lot more area for absorption to occur. With example A, I have a much decreased surface area. So that's the function of microvilli. Now, it's important to highlight really what's going on inside of the nucleus, because that is the driving force, the control center of what this particular cell is supposed to do. So our DNA, the full name for that is deoxyribonucleic acid. This is going to control what that cell is going to be doing, protein synthesis. And then we'll also want to divide up that DNA when we want to produce more cells. Say for like example, my
What do you mean you only have one leg? What happened?
nuclear envelope with nuclear pores in the envelope to allow for substances to get in and out. But when we look at the DNA, I'm sure most of you have noticed before, seen before, that we have this double helical shape. Well, basically, I got a long twisty thing, right? And anytime I have a long twisty thing, I want to kind of organize it so it doesn't get all twisted on itself, even at the molecular structure. So I got these histones, these proteins that the DNA is going to wrap itself around. So we create these nucleosomes, which when we are not going through that replication process, this is all unwound. If you look at this with an electron microscope, it kind of looks like it's fuzzy. You would see inside of the nucleus. If this is the inside of the nucleus, it would kind of look like this. Like there's just not any organization to it. And it's really pale and fuzzy looking. But then, right, so that's the chromatin. And then right before we're actually going to go through that division, that chromatin is going to condense, and that.
How long has it been since your leg was sorry, English is refusing me. How long has it been since your leg was amputated?
No, ma'am.
Okay. And we all know that humans have 23 pairs of chromosomes. And the last pair is what we call the sex chromosomes. This is a basic anatomy and physiology class, what we call a survey anatomy and physiology class. So we know a lot more now about, male and female.
Wait, you said 5 days? Are you still in the hospital right now as you speak?
X chromosomes, they are given the female title, and if an individual has an X and a Y chromosome, we have a male. And the other 22 chromosomes are what we would call our somatic chromosomes.
Oh my God. Did they catch the drunkard though? Because you said you were hit by a drunk driver. Did they ever catch him?
Body stuff. Now, there are a couple of other unique structures that we have to a cell, or could have with a cell. We don't always have this stuff. There's the cilia. And cilia are like hair extensions coming off from the plasma membrane. So, you know how my hair is like hanging off my head? That's what you want to think of the cilia. It doesn't have the actual components, the cytosol, deep to it like we see with microvilli. And cilia is going to be in a place, in an organ, on the top of tissue, where we want to move stuff across the surface of that cell. Propel substances, right? And the cilia will beat in one direction so that whatever is on that surface of the cell can move in the direction that the cilia is beating. So a perfect place that we would see this is like the respiratory tract, where we have goblet cells. And these guys are the cells that make.
How are you doing though? Like, how do you feel? Hi. Yeah, they did, and he got 5 years. I was in court with my mom, and said everything. That's great. That's wild. I do hope you feel better soon, like way soon. Do you think you'll be able to start school at the same time with, like, other people, other kids?
That can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough.
And the flagella is the last extra structure that we would need to address. And the flagellum singular, is going to be this extension that's coming off of a cell that's going to help the cell to actually be propelled long distances. So the only example of a cell in the human body that has flagella is going to be male sperm cells. So girls, sorry, you don't have any cells with flagella.
Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell.
See, so now you see I wasn't lying, because he said it too. Any questions with the organelles? I know that this is a busy unit, and I'm sorry. It's just in the beginning, there's just a lot of stuff. Make sure you study with not only the chapters, but the study guide as well, which is found in the syllabus. Starts on page 7. That's where you would see your study guide starting.
Okay, you said you were gonna get like a prosthetic leg. Okay. How do you feel though? Like about the whole situation? And also, are you still in a lot of pain, or like are they giving you enough medication for you not to feel much pain?
People that we are today. Plus, for example, any type of tissue that I have in my body that is going to need to be replaced, and basically all of it, or best example that we can think of or can visualize is the stuff that's coming in contact with a space, either the outside space or the inside space, like the GI tract. That stuff is going to need to regenerate itself pretty quickly. So there are two main parts of the cell cycle. We have interphase, and we have mitosis. Now, with mitosis, what essentially is occurring is we have a parent cell, and we create two identical daughter cells. So like I have a stem cell that's going to replicate and divide, and I'm going to get two daughter cells, and now I can replace my skin cells that are lost. When you get to anatomy and physiology 2, you'll talk about meiosis, which is occurring with the oocytes and the sperm cells, but don't worry about that for now. It's coming up later on. And in interphase, we're really trying to get ready for this division. So not only is it the DNA that needs to replicate itself, but also we need to make sure we have enough of the other components of the cell. Do I have enough cytosol? Do I have enough mitochondria? Do I have enough ribosomes? All of that needs to also be equally divided. And that replication process and really the preparation for division is occurring in interphase. The division of the genetic material is occurring in mitosis.
In this lesson, we'll be talking about how cells reproduce. How and why do they do this? Well, they use a process called cell division to make new cells, called daughter cells. Unicellular organisms, meaning creatures that consist of just one cell, such as bacteria, usually clone themselves during cell division. The two daughter cells that result are separate organisms. In this case, two new genetically identical bacteria. This is a type of asexual reproduction known as binary fission. Cells in a multicellular organism also reproduce by cell division. But the new daughter cells that are produced are not two separate organisms. Instead, these new cells are just parts of the organism, allowing it to grow or sometimes replacing cells that are worn out or injured. For example, your body heals a paper cut through division of your skin cells, occurring at the edges of the cut. In a modified example of cell division, sex cells called gametes are made.
It was scary. I got through it and I did not cry. I've had 12 shots and I have an IV drop. What is that sound? Like a drum sound? No, it wasn't a drum sound. It's I'm kind of taking like a little test thingy, but I couldn't figure out what exactly they wanted me to do. But I think I'm I'm figuring it out now. Shit. Me and English. But yeah, that's what the sound was. I kept getting it wrong because I couldn't figure out what exactly I'm supposed to do. But I think I got it now. Let me try again. Sorry about that.
Code can be passed on to each daughter cell.
Have your friends been like at the hospital to visit you?
Each side of an X-shaped chromosome is a genetically identical sister chromatid, forming a sideways V shape. In the middle, a structure called a centromere joins the sister chromatids together. Different types of organisms have different numbers of chromosomes. Every cell in your body is called a somatic cell, except your gametes. Human somatic cells have 23 pairs of chromosomes, for a total of 46. It's like the difference between how many pairs of shoes you have versus your total number of shoes. Gametes are the exceptions to this rule. Human eggs and sperm have only one chromosome from each pair, for a total of 23. And, unlike somatic cells, gametes are not genetically identical to their parent cells. When a sperm cell fertilizes an egg, they both contribute their 23 chromosomes. This fused cell, called a zygote, now has 23 pairs of chromosomes, for a total of 46. This overview of cell division will help prepare you for studying the cell cycle, which is the life cycle of the cell.
And yeah, you really don't get to meiosis until anatomy and physiology too. So
okay, that does make sense. But the good thing is school's about to start, so you'll see them there. Well, I'm glad you're okay now. You know, I'm glad that you're much better. It is going to be like a huge, drastic change, but you'll be fine. I don't know you, but from the way you've been texting, I can tell you're a nice person. I feel like you will be fine.
So maybe there was some type of mutation where that stop codon failed to actually control that cell division. And now a cell is going to divide uncontrollably. So, you know, a bunch of different things can cause this, you know, just a spontaneous mutation. Maybe it's an environmental factor, maybe it's inherited, you know, some cancers are inherited. So there's a lot of things that can cause cancer. We all know that there are many, many different types of cancer, and not all of them have the same, they don't have the same cause, but that's what cancer would be.
In this lesson, we'll be looking at the cell cycle. This is the lifespan of a eukaryotic somatic cell. A somatic cell is any cell in the body of an organism, except for sex cells, such as sperm and egg cells. The cell cycle describes the sequence of cell growth and division. A cell spends most of its life in a state called interphase. Interphase has 3 phases: the G1, S, and G2 phases. Interphase is followed by cell division, which has 1 phase, the M phase. Together, these 4 phases make up the entire cell cycle. G1 of interphase is sometimes called growth 1 or gap phase 1. In G1, a cell is busy growing and carrying out whatever function it's supposed to do. Note that some cells, such as muscle and nerve cells, exit the cell cycle after G1 because they do not divide again. A cell enters the S phase after it grows to the point where it's no longer able to function well and needs to divide. The S stands for synthesis, which means to make, because a copy of DNA is being made during this phase. Once DNA replication is complete, the cell enters the shortest and last part of interphase, called G2, also known as growth 2 or gap phase 2. Right now, it's enough to know that further preparations for cell division take place in the G2 phase. Now that interphase is over, the cell is ready for cell division, which happens in the
Hey Stella. Oh, I love you too. You sound really, really sweet. I've been in the hospital 12 times this month. Wow. You know what? If you've been in the hospital that many times this month, it most probably means you won't be there as many times next month. So what does that mean? It's gonna get better. I feel like it's way better you're in the hospital a lot this month. You get everything cleared out, you get healthy, you know? You get way better. By the time you go back to school, everything is like set. You know, you feel better. Are you gonna go for like physical therapy? I mean, have they told you about that or not?
Finally, we have cell division in the M phase. The M phase consists of mitosis, which is nuclear division, and cytokinesis, or division of the cytoplasm. We'll explore the details of mitosis and cytokinesis separately.
I'm so tired.
Let's get that out of there. So, interphase.
Oh,
Interphase again is where basically the cell is getting ready to do
24 minutes to go.
Interphase is definitely going to be the longest phase that any cell would have. We got to make sure that we've got enough cytosol. We got to make sure that we've got enough organelles. We got to make sure that we are prepped and ready to go for that one important part, mitosis, where the actual DNA that has already replicated is going to be able to be split into two. Now, sometimes a cell is no longer going to be going through the process of mitosis. It's not going to be able to replicate anymore. Or maybe just for a long period of time, it's not doing that. And if so, we would say that this cell is in the GO phase, where it's temporarily stopped dividing or resting, or it doesn't have the capability to do that. Like red blood cells, they're always in the GO phase. These guys can't divide. That's why red blood cells only last for about 120 days. But when we are prepping for that division, when we're talking about the components of the cytosol, so the organelles and the fluidy portion, that's going to occur in G1 and in G2. So G1, G2, we're duplicating the organelles, we're growing, we're getting more cytosol, and the same thing is happening in G2. In the S phase, this is where we are synthesizing more of the DNA itself. We're not splitting it just yet, but we're making sure that the DNA is replicated. Everything before mitosis.
Yes, I will. Okay, good. I'll be back. I've got to get... Okay, yeah, take your time. Take your shot. I'll still be here. I'm still going to be here for like the next, say, 25 minutes. And then I'll end the live. That'll be what, live for 2 hours? Take your time. I'll still be here.
Great, because we need to make sure that we are able to split the DNA. Those that chromatin is going to condense, and we're actually going to see the chromosomes now. Also, what you'll see is the centrioles are going to go to each one of the poles of the cell and start to create these long arms. They're called the mitotic spindles. Then we'll get metaphase, everything in the center, right? So metaphase, I've got all of my sister chromatids lined up. The centrioles are at each one of the pole, and those mitotic spindle fibers have reached out towards those sister chromatids. They're going to attach onto the center. I like to think about it as like the center belt, the centromere, so that when those mitotic spindles attach to those centromeres, now we can pull apart those sister chromatids in anaphase. sequence. So we've duplicated the DNA, we've replicated, we've doubled it. Now we got to half it, right? Because if I started at the parent.
Wait, you're asking if I'm gonna be on tomorrow? No, I won't be on tomorrow. This is my last time being live this week. I might go live again on Sunday. Yeah, I'm gonna watch them. Actually, I don't know. No, I don't know. I'm not sure when I'm gonna. I know for sure I'm gonna be live on Monday at 1 p.m. to 3:30 p.m. That's Dallas time. Dallas time is what, Central or Eastern? I never seem to understand. But yeah, I'm gonna be live at that time on Monday, and then on Wednesday I'll go live again from 1 p.m. to 6:30. That's a very long time to be live, but you know, do what you gotta do. Hey, let me read this name. It's called Fami. Hey, Fumi Lola. The name is Fumi Lola Akiniele. Please tell me if I'm pronouncing that right. Please, what's the live about? Nothing. The live is about nothing. Absolutely nothing. I'm bored enough. I'm in class right now, actually. And that's when I go live. I literally just put the live on and sit here doing absolutely nothing. Unless you guys ask me questions and then I'll interact, you feel me? But if nobody speaks to me, I don't really have much to say. Also, I'm in class, so I kind of have to focus. So, yeah, there's that. Layla said, okay, because I've got to go for a surgery. Oh my God, so good luck with your surgery. I hope it goes well. Thank you so much for watching my live. I love you. Bye. Yeah. For me, Lola, bro, I'm telling you, this live is about nothing. Like, I'm just here. I am in class though, so let me focus. I literally only come off to answer a question for like 10 seconds and then I go back. I kind of need this to keep saying, because if I don't do this, I'll be so bored in class. What are you studying? I'm studying nursing. I'm in an anatomy and physiology class right now. A lab class, actually. Yeah. Are you in school?
Opposite sides or poles of the cell. While this is happening, the nuclear membrane surrounding the nucleus disappears. Now that chromosomes are no longer separated from the cytoplasm, the opposite ends of the spindle fibers can attach to the centromeres. Next.
What's your major? Wait, no, first of all, which level school are you?
Line up the chromosomes along the equator of the cell.
Yeah. Bro.
The end-to-end.
I don't think I would survive. You said biotechnology? You got it. Hey. Just the word itself sounds hard. Are you guys like in holiday right now, or are you taking classes as we speak?
From each other, each chromatid is called a chromosome. The single-stranded chromosomes form a V shape as the spindle fibers shorten and drag them through the gel-like cytoplasm. The chromosomes move to opposite poles of the cell toward their centrioles. It's common to confuse.
Oh, okay, so it's nighttime. Do you have classes tomorrow? My bad, I'm looking at the computer so they don't realize that I'm actually not talking to them.
Reforms around each set of chromosomes. Then, the chromosomes spread out into chromatin, and the nucleolus becomes visible once again. Mitosis, the division of the nucleus, is now complete. The final step of the M phase is cytokinesis, the division of the cytoplasm. In animal cells, cytokinesis occurs through the inward movement of the cell membrane. This progressively pinches the cytoplasm until two identical daughter cells form. In contrast, plant cells can't pinch in two because they have a rigid cell wall surrounding their cell membrane. Instead, cell wall material assembles along the equator, forming a structure called the cell plate. The cell plate grows until it joins with the existing cell membrane, separating the two halves of the cell into daughter cells.
Oh.
Over time, new cell walls form between the two daughter cells. Here are the key points to remember. The M phase is the...
And we don't need to watch that again. Because I went through it, then he went through it, and then we're going to beat that horse pretty dead. How's that going so far?
I'm taking this lab class. Bro, let me just tell you, I'm over it. I'm over school. I'm over everything. But the only thing that keeps me going is the fact that I can't be broke. I can't be broke. Oh my God. I need to make like serious money. So the only thing that keeps me going is the thought of finally getting a decent job and getting me enough money to buy land. Yeah. I need to buy land. Like, not, oh, yeah, I need to get a house. I need to get a Birkin bag. Obviously, I want a Birkin bag. Obviously. But I want land more. Because land will buy me even more Birkin bags. Oh my, hi. Hey, how are you?
Until there's the same amount of particles on each side. Now, what I drew here was simple diffusion. Simple diffusion is saying that these particles will just simply melt across the membrane. It's pretty fast, right?
Mm-hmm.
But then we have facilitated diffusion, and that's a little bit different.
Oh, I love you too. Hi. How are you? By the way, that was meant for Arcane.
My integral protein that's working as a channel, and this is still a passive
Where are you watching from, Arcane? If I may ask, if you don't mind telling me.
Each individually, or maybe two at a time, or maybe three at a time, go through that door that's facilitating the movement. Still down its concentration.
Well, obviously you're watching from your phone or your tablet or something like that, but that's not what I meant. I meant, like, where are you located? Like, are you in Dallas like me? Because I'm in Dallas. Where are you? That's what I meant to ask.
Created by the mitochondria. And
so, like, for me, Lola is in Nigeria. Layla is in the UK. I never asked Stella where she's from.
Transportation will pump these few particles to the side where they're already in a high concentration. And then we have osmosis. Osmosis is exactly like simple diffusion, except for that membrane is permeable to water. So osmosis is the net movement of water.
Okay, your name is Lexi. Hi, Lexi. My bad. I was calling you Arcane because that's the name I was seeing up there. That's my fault. My fault, gang. Where are you watching from, though? Like, I don't mean your phone. I mean, where are you located? That's what I need to know. It's crazy that I've been live for almost 2 hours now.
Okay, if not,
like, what?
Have fun with Professor Bowen, and I will see you guys on August 20th. That's when I'll be back.
Yay.
I will be in the classroom starting on the 14th, but I won't see you guys until that next Wednesday. Enjoy the rest of your summer. Take care.
Safe travel.
Have a nice vacation.
All right, thank you.
Travel. Enjoy.
Thank you.
Thank you. It's gonna be fun.
Bye.
Take care.
I'm gonna send you an email concerning my week 1. Hello?
Hi, I'm here.
Yeah, some of my labs. The first week I had trouble with my computer, so I actually had to go on campus to get the iPad. That's what I'm using now.
Sure.
So
Yeah.
Okay, so it seems like class is done. Finally. Thank you, Jesus. Oh my God. Good knows I am tired of this class. No, I'm tired of school in general. Not even this class. I'm just tired of school. Yay! I'm free! Yes! Thank you, Jesus. Oh my God. Oh! Finally. Oh, bro. You don't understand. Wednesday is like my worst day of all. Like in the whole week. Wednesday is my worst day. Oh! But yeah. What state are you in? That was my question. My bad. I've been in class since Wednesday. You know, I have 2 classes. I have one that starts at 1 p.m. It ends at 3:30, and then I have another one at 4 that ends at 6:30. Oh my God! Actually, ends at supposed to end at 6:15. Right now it's 6:17. Anywho, it's fine. It's completely fine. So since we're here, what I'm gonna do is just finish all my quizzes for this class. That way I'm done with it. I know I don't have to come back here again. Period. Lord knows. God knows I'm tired. Okay, Lexi. Thank you. I will keep going. I mean, I literally have no choice. I've paid way too much money to get here and just quit. Lexi, you're in America. Okay. At least now we're going somewhere. But what state are you in? Like, oh no, my light is battery low. Oh my God. Something's always happening. Something is always happening. But you know what? The good thing is I'm already done with class, so I don't really need the lights. Plus, you guys can see me, so yeah. Oh, ghost. Ooh. Don't mind me. I'm quite silly at times. You're in Arizona. Oh my God, that's hot. That's hot. I have a cousin who lives in Arizona. I could never survive in Arizona, bro. I would die from the heat alone. No, thank you. Okay, D is... I need you guys to help me out with the homework, since that's the only thing we're doing now. Let's do it together. Come on, stick. Let's go right here. Oh, okay. I don't think I'm supposed to be exposing that. But anyway, I'll show you guys as I do my homework. Let's go. So D is interface. One second. Okay, let's go. Interphase. And then the next phase is when they connect, which is metaphase. That's E. Oh, you're not seeing. You can see your screen. Yeah, I'm pretty much done with class, and I want to get this done. So yeah, D is interphase. The next, there's supposed to be the next stage, which is metaphase. And then when they separate, that should be telophase. So this. And then after telophase, we have wait. Have I made a mistake? I think I made a mistake. I know this is interphase for sure. And then wait, now I'm very confused. Very confused. I think this is interphase. This is interphase. B is interphase. Freaking idiot. Okay, B is interphase. Then from interphase, they what? They connect, which is E. That's supposed to be prophase. So E is prophase. And then, no. E is metaphase. Okay. I know anaphase is A for sure. When they're like separating, but they're not separating. That's A. And then C is telophase. What's D? One, two, three, four, 5. Why are there 5? We have B as interphase. We have E as metaphase. There's no prophase. That's what's confusing me. That's what's confusing me. No, but B can't be prophase because prophase is when they like come together, right? Before they separate. That's prophase. Also, oh, that's the You see how God is just wonderful. Okay, so now let's start again. Let's start again from the beginning. Yeah, let's start from the beginning, please. Yeah, B is obviously number 1, which is interphase. B is the first one, interphase, before anything comes together. And then the second one is supposed to be prophase. Prophase is when they've come together, but they're not really separated. They're just together. So which is prophase? Oh my God! I'm sorry. I didn't mean to yell. I'm just frustrated. I think E is prophase. I mean, they're together, but they haven't separated. They just joined. So I think E is prophase. And then metaphase is E. Metaphase is E. Oh. Anaphase is the separating, okay, which is A. C is obviously telophase. Why are you guys moving? Wait, what happened? Oh, okay. Prophase is the second stage. Yes, prophase is the second stage, but I can't seem to figure out which one it is. This image is not clear. B is interphase, step 1. I can't seem to figure out which one is prophase. So let's skip. Let's go to metaphase. Metaphase is when they have these little lines joining together. So that's clearly metaphase. Prophase, chromatin. Yes. That's when the nucleolus is supposed to disappear. You're right. You're right. D is prophase. Wait, you said B? Wait, if B is prophase, then which one is metaphase? I think that's why I'm confused. I'm clearly confusing metaphase with prophase. So, but come on, don't block my screen. I don't have patience for this. Okay, let's go. Well, you're not seeing the whole screen. There we go. Now everyone can see my screen. Okay. Let's relax. You know. Is there a way to clear this? There's no way to clear it. Okay, interesting. E is metaphase for sure. E is metaphase for sure. D is interphase and B is prophase. So D is interphase, so I don't need to change this. Prophase is B. Okay. Next is supposed to be metaphase, which is clearly E. Then anaphase is when they start this little separating thingy, but they still, okay. And then telophase is when they are very close to separating, which is C. We just don't have cytokinesis on this one. So this is correct. Yeah, we're good. Yeah. Great. Yay! W for me, Lola. Thank you very much. Thank you for coming through. You're a real one. Love gang. All right, let's go to the next one because I'm tired. Okay. Submit. Loading. Yay, we got it. Come on now. Okay, this is fun. I like this. Let's go. Let me turn this. Return. Let's go to the next activity so we can get done with this very quick. Kind of need to pee. I don't want to pee before I finish the live, but I'm actually dying. Okay. Diffusion and osmosis. Describe the process of diffusion and osmosis. I already did this one in the other class. Practice worksheet. Thank you very much. I appreciate it. Oh, come on. Hurry up, please. Please. Okay, we're about to finish. Let's just take these and go. Which of the following factors influences the rate of diffusion that takes place? Temperature, pH, Size of particles, I think so. What the hell? Huh. I don't like seeing that, so I'm going to take it. I need to have 100 on every single thing. It can't be failing. Temperature, pH. Eh? Wait, okay, now I'm confused. First, wait. First, you all saw that, right? We started the first round. How is it that just relax, it's calm, it's calm. It's calm, you're done with class, it's Wednesday. It's just another one here to support you. Hi. By the way, the name that I'm seeing is God's Greatest Child. That's a very bold name, you know, considering that I'm God's greatest child. My bad. We're all children of God, and in his eyes, I believe we're all great. I can't say anything from God's perspective because he's God and he only knows. It's just an assumption. But yeah, that's what's up. Okay, let me try and finish this because now this is just pissing me off. Incorrect. Okay. Let's take this again. If it fails this time, I'm done. I'm done. Like I'm finished. Like finished. Like done. Like out. Like. Okay. So size of particle, temperature, stiffness of the gradient, whatever. Submit 100. Yay, we're done. Next. No, I'm not going through that. I'm not studying that one. I don't have the energy to study anything else. Begin. Loading. You're loading a bit slowly. A hypertonic solution is osmotic concentration is dissolvent causes water to move. Bro, where did I go? Hypertonic and hypotonic. Okay, so let's read again. So hypotonic is when it allows water to come in. A hypotonic solution osmotic concentration is higher than the cytosol and causes water to move into the cell by osmosis. Submit 50. Why? What? Oh, hyper. Ugh. I thought it was hypo. Then how did I get the first one? Huh. Very weird. Okay, let's take it again. Hypertonic solution is higher than this because it's out of. It's hypertonic, not hypo. Crazy. I appreciate the support. I really, really do. Return. I really want to finish this like today, like now, like I don't want to come back here. Osmosis experiment instructions. Bro, are you telling me we have to do this? Please don't tell me we have to do this. Select experiment either eggs or gummy bears. If not using eggs, skip to step 4. Oh my god, we have to do this. Oh, no. Why? Oh. I don't think it's graded anyways. Oh, you can't see my screen well. My bad. Let me fix that for you. Wait, let me first clean my camera. It might be the issue. I wish there was a way to like flip it, but from my understanding, the only way to flip it is if you do it before you start the live, and I already did it to, you know, be like this. I can't flip it. Like if I do, what? No green screen. I beg. Okay, cool. Let me not sing. Next thing I know, they're telling me that my 2-hour plus video live is copyright and they're not gonna count the hours. Yes, it has happened to me before. That's how I learned. I learned my lesson the hard way. Yeah. Never again. Okay, let's go. I hope you can see my screen better now. What an unfortunate situation. I'm tired of doing things. Okay, that seems pretty clear to me, at least. It's not focused though. Wait, I'm trying to focus it. Boom. Boom shakalaka. There we go. Okay, now that's, I kind of don't want you guys to see that info up there. Okay, I hope that covers most of it. Let's go. The claim in the photograph before has been dissected language. I've done this. Why am I doing this again? I don't understand. What is this supposed to be? I don't understand. Why am I doing this again? That's what I need to understand. What an unfortunate. Fuminola, can you see my screen now? I hope you can. Let me see. But I'm trying to find something and I can't seem to find it. It's starting to piss me off. Not at all. What? How? How come? Okay, well, let me just, my bad. I'm sorry you can't see my screen. I've tried everything I could possibly think of. Well, I could see my screen on my phone. I'm just not sure why you can't see it. Sorry about that. Let me just put this over here so I can take this test. Is it timed? No, it's not. Okay, good. So I think it's photograph. I can't seem to find this. Why does this keep going off? It's not even like... Okay. Was it not charging? Like, bro. I don't understand you. Can you, like, not stress me out, please? Is it? Don't tell me that you're dying on me. Okay, it's charging. It's charging now. It's charging now. I wasn't charging the whole time. That's so annoying. But, oh well. Why you gonna do like? Let me relax. Sagittal in the image below. I can see, but not the full screen. It's okay. It's okay. Let me just, put it in the way that you can see at least some of it. That's better than, not saying anything. Because this is actually a test. It's not just, the practicals that I was doing. So I kind of need to focus with this one, which means I won't be able to talk a lot or even look at my screen a lot. So yeah, I'm about to go like real quiet. Okay, that's much better. We're looking down into the patient's skull from above. Which of the following describes this image? Can this thing like quit adjusting itself? I need it to just be there. Oh my god. But can you like quit stressing me out? Thank you very much. Much better. Okay. That's the inferior view, because inferior is looking down, and this is obviously a transverse section. Definitely not sagittal. That's crazy. What section of the clay man will yield the medical image shown below? This section of the lab assignment refers back to Unit 2, specifically to the rise in pH strip activity or whatever. Okay. For those of you who don't know, the lower the pH, the more acidic, the higher, the more alkaline. pH testing. What exactly am I supposed to be doing? Multiple choice, point 1. Look at the image above, captured from lab. Sample of drain container shown to leave a very high concentration of blue hydroxide ions, so base, and a very low concentration of red hydrogen ions. Okay. What can we conclude about the drain cleaner pH? It's basic. Image above shows a pH test strip. Okay. What is the pH of the human blood? Wait. It's like a 7.4. It's closer to this and this. What's the function of a buffer? Select to all that apply to act as either. So needed to help maintain pH balance, to either donate or accept a proton from the solution when needed. Yep. Base, acid buffer. Which beaker will require more base to be added? Wait. Pour here, balance. Pour here. Yeah, pH of human blood is 7.4. So that means that blood was actually stable. Exposing the pineapple to poor conditions, causing the protease enzyme to unfold and lose their 3D structure. This made them unable to catalyze chemical reactions anymore. What word best describes this situation? Exposing the pineapple to poor conditions caused the protein enzyme to unfold and lose their 3D structure. This made them unable to catalyze chemical. They denatured. Yep. Pineapple containing enzyme breakdown of proteins. Functioning protease will break down the proteins in gelatin, turning the gelatin into liquid. What should happen to gelatin if added to a protease enzyme that has been exposed to high heat? Nothing about the gelatin will change. The gelatin will liquefy. The gelatin will start to liquefy and then turn to normal. The gelatin will change the function of the enzyme. It will not change the function of the enzyme. There's literally almost no enzyme left. I feel like nothing will happen to will change. What are the requirements for enzyme to function efficiently? Proper temperature, proper pH, proper generation. This section of the lab segment refers, I don't care what it refers to. In this demonstration, we saw that the salt tablets readily dissolved in water while all droplets flee from the water refusing to mix. Categorize each word as hydrophobic or hydrophilic. Every word should be categorized. Do not leave any words uncategorized. Okay. Hydrophobic, water fearing. Charge oil. Hydrophobic. Water loving, hydrophilic. Salt, water loving, hydrophilic. Repel by water, hydrophobic. Attracted to water, hydrophilic. Bulky. Now, this is the part I don't remember. Is it polar molecules that love water, or is it I don't remember at all. Let me see. Are polar molecules hydrophobic or hydrophilic? The image above shows a pitcher of water with sugar added. There is a precipitant at the bottom of the pitcher, which could make more sugar dissolve. Add acid to the solution, add base to the solution. Make the water cold, heat up the water. Above is an image of salt. Match each component to the solution. Salt water is the solution. Oh, I need to pee. Anyways, salt water is the solution, water is the solvent, and this one is a solid. If an egg was placed in a hypertonic solution, okay, the water would smooth out of the egg. Ouch. Wait. If an egg was placed in a hypertonic, which means it doesn't have water, so the water is going to get out of the egg. This would cause the egg to shrink or shrivel up. Control egg A. Egg A has been replaced into a syrup solution to the left of the control egg. It has been treated based on the results experiments. The syrup is hypertonic, or water got out. Syrup is hypotonic. Let's read it again. Syrup solution No, it's hypertonic. No, it's hypo. Wait. Okay, now I'm confused. Wait. Let's first relax. Yeah? Let's first relax. Cause. Wait a minute. Hypertonic, hypo, makes the cell swell. Cells swell. This one shrunk, so the egg is hypertonic. That means the solution was hypotonic. It had more salt than water. That's why it absorbed the water that was inside the egg. So it's hypotonic. If an egg was placed in a hypertonic solution, if an egg was placed in a hypertonic solution, so basically a solution with more salt and less water, the water would go out of the egg. This would cause the egg to shrivel up. Okay, now that this egg has shriveled up, it doesn't make sense for us to say it's what? Hypertonic, because the solution was hypotonic. It had more water, so it went in. Okay, let's go. Is that it? Am I done? Please tell me I'm done. Please tell me that I'm done. What's the next one? Oh my god, that wasn't even the quiz. Oh my goodness gracious. Hi. My light is not even charging. Bro, can you charge? For the love of God. Thank you. That wasn't even the quiz. I just wasted my time. Anyways, you know what? Let me this quiz is actually gonna be timed, so I'm gonna leave you guys. Hi Ray. I'm sorry, but you've come when I'm actually ending the live because I'm actually tired. Not that I'm tired. I'm gonna take this quiz. I only have 12 minutes to focus on it. How many questions do I have? I have 10 minutes. Okay, cool. So bye for now. Love you guys. Love. Gang. Bye. Mwah.